we're still struggling to get the outcome data. But what we currently see is that if we apply the severity-based proteome signature of central carbon metabolism changes in liver cancer, and we apply a machine learning approach, it enables us to predict the disease course of precancer and cancerous conditions in the future. So with that, in 2017, so basically, I got my thesis and a couple of papers published on that, and another one is upcoming also. And I had a side project also studying the metabolome of gut. And this paper got published in Nature, which was pretty amazing for me to have a side project flying so high. It was a nice experience. So after that, I moved to Andrei Shevchenko lab in Dresden, in Max Planck for molecular cell biology and genetics. And again, I was looking forward to do the translational research and have some project management experience as well. So I, again, focused on precancerous conditions in liver and working with mouse and human, mainly with lipidomics, and also applied lipidomics to laser capture microdissection, and yeah, a little bit of proteomics. And this study is currently being finalized. So basically, we applied this shotgun lipidomics to around 400 samples, human biopsies, and we figured out certain things, such as, for example, that fatty liver is not just neutral, simple fat accumulation, and then there's particular sphingomyelins involved in fatty liver development, and some changes depend on mutation, and there are some changes which are independent on mutation, but still we have to figure out what is the underlying cause. Part of this cohort has already been published this year in gut. Then we also have published a review on plasma lipidomics, and also shotgun combined with shotgun lipidomics combined with laser capture microdissection has been also published. And now we've got a new grant for 3+2 years. And currently I found myself in the situation where I'm pretty comfortably looking for a new project management position, because I have a possibility to stay as a postdoc for extra 5 years. So the project management in academia so far I've been doing is planning of the experiments and further analysis, establishing and maintaining collaborations with informaticians, bioinformaticians, medical doctors, and biobank folks, establishing, which was pretty challenging, a proper communication between all members from 5 labs, performing regular meetings and so on, and also establishing and overseeing all the milestones for this project. And I've been also doing pretty decent amount of paperwork for this project. I also have established a campus MS community in Dresden. We're having regular journal clubs, external and internal communication I also promote. And we've established annual meetings, we call them FeMS updates for Christmas. They're pre-Christmas, and unfortunately this time we'll have to cancel one, but it's been pretty cool since we got people from Germany traveling to our place and, you know, got a glühwein and discuss science. It was pretty cool. I'm also taking care of the webpage and of the clinical lipidomics project and so on. And also currently I'm getting a project management education, the distant one, in one of the universities in Germany. And I have also been involved in some project management outside of academia, mostly focusing this on mental health advocacy in academic environment. And I have established a mental health initiative locally, including mental health first aid program, founding experience postdoc program, postdoc pre-doc tandem, lectures and seminars, which was one of the biggest achievements which I consider of my efforts, is that we have established the mandatory seminars in supportive communications for PI research group leaders and faculty, in facility group leaders. So this was one of the greatest achievements, I think, of this local initiative. We have also organized the one in Max Planck Society, which includes more than 80 institutes all over Germany. And we have, this year, we have ran the fully online mental health awareness week with more than 1,000 attendees, which was pretty cool. And globally, we established with a bunch of other volunteers the Dragonfly Mental Health, which is promoting an excellent mental health and fighting stigmas and driving the systemic change in academia. So I'm there, I'm an organizational team lead. So I take care of everything when it comes to the volunteer communication, lecture seminars, internal communication, webpage, and so on. And with this, I decided that since I really enjoy the project management work, I would like to move to project management. So I'm particularly looking for the project management position in translational medicine, whether it's academia, pharma, or certain startups, it doesn't really matter to me. I just, I love promoting the communication between partners with different backgrounds, and I'm really good at it. And I think this is my strong spot, and I would love to be doing this for my future career. So I'm good at translating the results of the scientific and medical work to the general public as well. And I mean, I just love organizing small and things and large things, and I like doing it. And with this, I'd like to stop and thank you so much for your attention. And I'm happy to answer the questions if you have any.
Thanks, Olga. Sorry, I was having trouble finding my button, but that was great. So if there's any questions, go ahead and unmute yourselves. For Olga, I have a quick one to get started. And so I noticed a lot of your papers were on lipidomic studies, and so, or several anyway. So when you transition to more of a project management, do you have an omic that you want to be working in, or more of just like a clinical aspect?
I am particularly like doing multi-omics. So if this includes combination and integration of multi-omics data, that would be just perfect. I did it for my PhD. I know all this like side, I mean, so ups and downs of all these methods, basically. And I think I'll be really good at translation. And also I'm good at, so basically I can explain the limitations of each and every method, including the ones which medical doctors have. So yep. I've been doing this a lot during my PhD and my postdoc, and this is something which is really the value of it is underestimated, in my opinion, in the field.
Definitely. And do you have a location, or are you open worldwide?
So I am open worldwide. However, at the moment, I'm in the process of getting the German citizenships, or hopefully to be soon one. So as soon as I get one, the paperwork should get much easier for me. Because last time when I was working in the States for a year, it was difficult to get the paperwork done for an immigrant. And with the German passport, things are easier.
Yeah, that makes sense. Go ahead and unmute yourself, Jenny.
Oh, okay. Sorry. I just like to type better than to say. So I was just wondering, what do you consider to be your best strategy when you have to bring together these teams for the large studies that involve the biosampling team, the clinical team, which are often nurse coordinators, the technology, and the bioinformatics? Do you keep them together, or do you separate them and only bring them together sometimes? What's your strategy?
So it depends from case to case. Sometimes you need to bring people together, and I believe in separate meetings which should be combined, or like, so a couple of separate meetings and then all together meetings. And sometimes what I've noticed is the simple phone call works better than 20 emails.
Yes.
So my strategy is to call, or just to knock on the door when it's available, because otherwise, and you know, I mean, it's always nicer to talk to a person anyways. So you just organize small meetings if it's needed, and then like annual or weekly or monthly, whatever. So when we were working on the data analysis of this large cohort, which I'm currently working on, we had meetings every 2 weeks. Before that, of course, there were multiple smaller communication in between different members, of course, but then every 2 weeks we had a call where we were keeping updated each other on what has been done. So, I mean, and if there are some larger project, then of course you need some tools, Slack channels or Rocket channel or whatever it is. But yeah, that would be my way to go.
I definitely need tools. I don't think they always teach you all those tools that you need to organize.
This is actually very interesting. So I've been like doing my research when I was doing this project. And then I started to do this project management course, and there are a list of the things you may use. And I was like, I spent like weeks looking for that. And then when you go and do the course, everything's there. So yeah.
Yeah, I really wish that I certainly wish I had been taught those skills early on. It would have saved me a lot of time. So I'm really glad to hear you're doing that.
It was still very pleasant that, okay, in the principle, I did everything right, but a lot of things could have been simplified if I would, if I would have had this education or course as a part of my education.
Yeah, totally agree. Well done.
Definitely. Any other questions for Olga? If not, let's give her another round of applause. That was great, Olga. And we'll
thank you so much.
And we'll hand it over to Sitora now. So I'll keep mixing you guys up as we go.
Okay, good morning, everyone. Can you see my slides right now? Okay, let me see.
Yeah, we can see them. They're not in presenter mode, though. There you go.
Okay, so good morning, everyone. My name is Sitora. I'm from NC State, North Carolina, and the title of my talk is Fundamentals of IR-Maldacity Mass Spectrometry Imaging for Applications in Metabolomics and Lipidomics. So a little bit about myself. I was born in Tashkent, Uzbekistan, right here. I finished high school there, and then in 2012, I flew to Buffalo, New York to pursue a Bachelor of Science in Chemistry with a minor in mathematics. I also did an undergraduate research under Professor Frank Bright. In 2016, I decided to move to North Carolina, Raleigh to work for Professor Muddiman, where I did my work with IR-Maldacity mass spectrometry and imaging system. In my free time, I like to hike, I like to cook, and I like to read. So today I'm going to talk about two most exciting projects that I worked on in graduate school. These are spectral accuracy and direct analysis of bone samples. So a little bit of background. So mass spectrometry imaging is a technique used in mass spectrometry that helps to visualize the spatial distribution of different molecules in a label-free manner. So in a typical mass spec imaging experiment, one spectrum is collected from each spot on the sample, and then using mass spectrometry imaging software, we can generate heat maps which show spatial distribution of detected ions. So in graduate school, I've been working with IR-Maldacity system, which stands for Infrared Matrix Assisted Laser Desorption Electrospray Ionization. So this ionization source consists of ESI, electrospray, IR laser, translational stage, and mass spectrometer. So in a typical IR-Maldacity experiment, we first slice soft tissue, and then we form a very thin ice layer on top of it. Then we use IR laser, which desorbs primarily neutral species, which are then ionized in ESI plume. So IR-Maldacity has several advantages over existing imaging sources. So first of all is that ice is the only matrix that we use in the system. IR-Maldacity can generate singly and multiply charged ions. The system has very high tolerance for salt. In fact, we can detect lipids from pickles. And finally, we can absolutely quantify analytes from some of the samples. So now I'd like to switch gears and talk about the first project that I worked on in graduate school, which is called spectral accuracy. So spectral accuracy is the ability of a mass analyzer to accurately measure not only mass to charge ratios, but also relative abundances of isotopologues. So isotopologues, as you all might know, are molecules that have different isotopic compositions, and isotopes have different number of neutrons. So shown here is a representative isotopic distribution where we have a monoisotopic peak, which is composed of the most abundant isotopes, and then the first isotopologue to the right has at least one extra neutron. So knowing that the mass difference between these two adjacent peaks is 1.0034 daltons, we can confidently say that this first isotopologue represents a molecule with one carbon-13 in it. But why are we so interested in isotopic distributions? Well, accurately measured relative abundances in isotopic envelopes allow us to estimate atomic composition in a known molecule. For example, in this case, knowing that abundance of one carbon-13 on Earth is approximately 1.1%, we can estimate number of carbons from this isotopic envelope, which gives us approximately 8 carbons. So now I'd like to explain you how exactly we use isotopic distributions in untargeted mass spectrometry imaging analysis. So in a typical untargeted MSI analysis, we collect MS1 data from each voxel, and then we use imaging software to generate heat maps for all peaks of interest. The next step is to choose one heat map that shows a very interesting spatial distribution. So in this case, I chose peak at 154. Then we search this M/Z value against a database like Metlin, which later reports a very long list with M/Z values and formulas. But the problem here is that usually one M/Z value has several formulas that correspond to the mass. So to reduce the list with IDs, we can use isotopic distributions to estimate number of atoms from spectrum. So this is just an example. So here I'm showing isotopic distribution for a molecule at M/Z at 154. So this is this heat map. And so using relative abundances in isotopic distribution, I calculated approximately 8 carbons. And so based on biological information and carbon counting, I could confirm that the peak at 154 represents a dopamine. So this is how we use spectral accuracy in untargeted mass spec imaging analysis. But the problem is that abundance of a peak from each voxel will be different. So therefore, in this example, we're getting 7 and 14 carbons from one of the most and least abundant voxels. So my job in this project was to establish abundance thresholds above which carbon, nitrogen, and sulfur counting is accurate in IR-Maldesi mass spec imaging. So to determine abundance threshold, I worked with 3 different molecules, caffeine, MRFA, peptide, and ultramark, which represented small, medium, and large molecules. So what I did first is I analyzed these 3 molecules using Q-Exactive plus mass spectrometer, and I recorded data at different ion populations or different AGC targets, meaning that I was trapping a different amount of ions in the orbit trap mass spectrometer to alter abundance of different isotope logs. Next, exactly the same standards from exactly the same lot numbers, we analyzed using isotope ratio mass spectrometer, which very accurately measured abundance of each isotope in a molecule. So knowing very accurate abundances of isotopes in each of these standards, we were able to accurately estimate number of carbons in each of these standards. Again, I'd like to highlight that using the isotope ratio mass spectrometer in this study was very crucial because abundance of carbon-13 is not only different from standard to standard, as I'm showing here, but it's also very different from IUPAC's best measurement value, which we oftentimes use for carbon counting. So these are the results. After estimating number of carbons from each of the standards, what I did next is I calculated the difference between known and observed number of carbons shown here. And then I found the optimum number of ions that result in carbon counting with a tolerance of ±1 atom. And then I did exactly the same analysis for nitrogen and sulfur peaks. So this table here shows abundance thresholds for 3 different isotope logs, carbon-13, nitrogen-15, and sulfur. And the last step in this experiment, in this study, was to just confirm that these thresholds are actually working. So what we did next is we looked at the mass spec imaging data. So this heat map shows abundance of glutathione ion detected from healthy and cancerous hen ovaries. And these are abundance values for sulfur peak. So the next step was to estimate number of sulfurs in each of these tissues. And heat map on the bottom right shows a number of shows a difference between observed and known number of sulfurs in each of these samples. So as you can see from the heat map on the bottom right, sulfur counting is much more accurate in cancerous tissue compared to the healthy tissue because abundance of sulfur peak in the cancerous tissue was above reported threshold. So this wraps up my first big project, spectral accuracy. And the last thing I wanted to talk about is mass spectrometry imaging of bones. I wanted to talk about it today because for me, personally, this was a very fun project to work on. So mass spec imaging of bones. So one of the conventional methods of preparation of bones for mass spectrometry imaging is decalcification, where fresh bone is soaked in various solvents for days and sometimes even weeks. So during this process, all minerals are removed from the bone, making bone very soft and therefore very easy to slice in thin sections. So today we have only two MSI sources that are compatible with mass spec imaging of bones. These are MALDI and SIMS, and both of these are done on decalcified bones. However, decalcification has two big disadvantages. First of all, it causes physical changes to the bone because you soak bone in decalcifying agent for days and weeks. And then a second point is that decalcification causes chemical changes to the bone, meaning that it can remove different molecules and extract different molecules from the bone. So in this work, we're able to develop a method for direct analysis of undecalcified bones using iron maltese mass spectrometry imaging. And in this method, we're not including decalcification steps, so meaning that we're analyzing a fresh bone. And then this work was also highlighted on the cover of Analytical Methods. So during method development, I came across two big problems. First of all is that slicing a fresh bone using cryomicrotome is almost impossible because bone tends to curl and just break apart. And then an alternative method for analysis of bone would be slicing a bone in half. But in this case, the flat surface of the bone will never be perfectly parallel to the stage of the imaging system, resulting in a not very even sampling of the tissue. So in developed method, what we do is we extract bone from an animal, we freeze it, we slice it in half using cryomicrotome, we trim it in half. Then we flip the bone and we embed it in an embedded material. And after approximately 15 minutes, we remove the bone and we flip the sample. So what we get at the end is a very flat surface of the tissue. And then we use imaging system to raster the sample from left to right, and then left to right. So during this method development, I tested these mini embedded materials, and the winner here is plaster of Paris, which satisfies all three parameters of mass spectrometry imaging using iron maltese. So it doesn't modify the bone. Plaster of Paris can freeze at very low temperatures, and most importantly, it does not suppress ESI signal. So at the very end, I analyzed two different bones extracted from a mouse that had a stroke and a healthy mouse, and then we putatively identified peaks using Metaspace software. So in conclusion, I talked about how isotopic distributions help with identifications of unknown in untargeted MSI analysis, and the second topic was about analysis of bone. And here I showed that iron maltese is compatible with direct analysis of bone tissues. At the end, I'd like to thank everyone from the Muddiman group, Dr. Muddiman, two people who trained me several years ago. These are Dr. Manzakelov and Dr. Milad Nazari. And finally, I'm looking for a job currently in industry, and I have a green card, so I don't need a work authorization. And my defense was scheduled for December 3rd, so I'm super, super excited about this. So if you guys have any questions, please let me know right now.
Thanks, Sitora. So everybody give her a big virtual round of applause. And does anybody have any questions for Sitora? Go ahead and unmute if you do.
No, no one has questions. That's really sad.
You might get away free.
No, so you can't get away free.
No,
so.
that's not allowed.
Someone needs to ask a question. So,
Wait,
so I was actually super intrigued with your choices of how to do the bone. I thought, I mean, that's really smart, right? What you've done. But I was also really surprised you got to Pasteur Paris. How did you go through that? Like, why did you think of all of those different matrices, including what you used to like do molds for dentists and stuff like that? So just super curious how you kind of thought through, or was that like a brainstorming in your lab, or how did you get there? Because that's a leap, right? Just curious.
Yeah, so basically initially when my job was to just analyze bones, right? And so initially we were going to work with very thin bone sections, which we could probably do, but and we still could, you know, detect ions from very thin slices. But the thing is that we were actually missing a lot of samples from that thin section because our cryo-microtome didn't have a diamond blade. So then I tried to embed bone in agarose, I think, but agarose you have to keep warm.
Yeah.
So that I didn't like because I could tell that bone was changing the color, and so I could tell something was happening. It almost looked like I was cooking the bone in the lab. So then I was like, okay, yeah, and so then I was like, okay, I probably need something else. I tried to glue the bone, which didn't work because it's glue. And then when I was buying the glue, I was buying the glue from Home Depot because it's really close to my home. And then when I was walking through the aisles, I started seeing all of these interesting embedding materials like thin set mortar. And then so I started buying all the stuff from Home Depot. And then
that's fun.
Yeah, it was really fun. But people from the business office at my department thought that I was using Professor Madiman's money to like
craft at home.
Yeah, to craft at home. But yeah, but it was just very spontaneous. I told Dr. Madiman about thin set mortar, and then I was actually seeing a dentist at the time, and then I was,
use that.
Then that's how I came up with alginate. It was just very spontaneous, very random.
Nice.
Yeah, I was just trying to think about an embedded material, and actually thin set mortar was recommended by Dr. Madiman, I think.
So when you were talking, I was imagining you out like doing dinosaur bones, you know, TRX, you know, these massive bones.
Oh, yeah.
I wonder how you'd pour the Pasteur Paris for that one. Congratulations.
Thank you.
Yeah, I love it. Your PhD thesis was inspired by Home Depot.
I know. I should probably acknowledge this. This is a really good point.
Yeah, I can't wait to see that in a week or two. Any other questions for Sitora? I think in respect to time, we might just get going anyway. Thank you so much, Sitora. So everybody, please give her one more round of applause and we'll head on to Tessa.
Okay. Oops. All right. Thank you guys for having me to talk today. I'm excited to share with you some of my dissertation research in the Hicks group at UNC Chapel Hill, identifying novel antimicrobial peptides. And I want to talk about some of the different methods that we're using the plant species Amaranthus tricolor as an example. And just before we get into that, a little bit about myself. So I'm from Bradenton, Florida. So our claim to fame right now is we were home of the WNBA bubble. And we also have the Tropicana orange juice headquarters. So lots of big trucks carrying lots of oranges. And then I went up to Tallahassee to go to Florida State University for undergrad, and I majored in chemistry and anthropology. And I was really lucky while I was there. I did my undergrad research in Dr. Marshall's lab at the National High Magnetic Field Lab, and I focused on characterizing different crude oils. So then in 16, I guess, I moved up to Chapel Hill and I've been working on my PhD in Leslie Hicks's lab. And so we have a focus in the portion of the lab that I work in on studying antimicrobial peptides, and that sort of the driver of this research is the antibiotic resistance crisis. So in 2019, the CDC estimated that about 3 million illnesses and 36,000 deaths each year in the US alone are associated with antibiotic resistance. So this really threatens our modern healthcare system, and there's a need for novel therapeutics. So we look at antimicrobial peptides as one possible natural product that could help alleviate this. So they're expressed by all forms of life, but today I'm going to focus mostly on plants. They're host defense peptides with a broad range of activities, so they can be antifungal, anti-cancer, antibacterial, antiviral, and they have certain common characteristics. They tend to be short, highly basic with a hydrophobic region. They're often cysteine rich, and those cysteines can form disulfide bonds, which help with thermal and proteolytic stability. And they can have a lot of other post-translational modifications, which make them really interesting analytical challenges to characterize. So in the Hicks lab, the majority of the time we focus on antimicrobial peptide discovery from plants. So we've looked at a wide variety of plants. So we've looked at a lot of different viola species. Here's my mouse. And those are traditional medicinal plants, and they produce some really cool cyclic peptides. We've looked at tobacco species, so more like model species like tobacco or tomato. And today where I'm going to focus is on this plant species, red spinach. So I looked at this plant species, red spinach, using two major methods. So the first is our FeMS pipeline, which the lab has developed. And so real quick, the steps of that are we extract our plant tissue into a crude extract so that we have all the peptides, and then we fractionate it. And then we screen the fractions of that peptide library against different pathogens to find interesting activity. And we'll profile the abundance of peptides across our library fractions using LC-MS, and then use statistical modeling to correlate the bioactivity measurements with the peptide abundances, and then identify most likely contributors to activity. So my work has focused mainly down here on this identify or to characterize likely contributors to activity. And then the other half of my research, which I'm not going to focus on as much today, has been looking at the mass spec data that we're generating as we're looking at all these different plant species and asking ourselves, is there an additional way we can mine this data? So can we predict likely antimicrobial peptides based on what's known about different classes, and then use a more bottom-up approach to identify them in our peptide extracts. But for the sake of time today, I'm going to focus on this first portion. So as I mentioned, today I want to talk about Amaranthus tricolor. So this is Amaranthus tricolor, or red spinach, and it has this really cool like 2 phenotype look when you grow it in the greenhouse. So you get some red leaves and some green leaves, which I just think is interesting. It's a traditional medicinal plant, but it's also edible. So sometimes you'll find it in the ingredient section in Trader Joe's granola bars, I've noticed since I started this project. And the peptide library from the aerial tissue of Amaranthus tricolor was active against E. coli. So here are the fractions of our peptide library. And then this is percent activity. So a higher percent activity means that the fractions could kill more cells. So this region here from about 30 to 35 was really active. We wanted to know what was causing it. So we profiled our fractions using LC-MS and used our PepSavvy-MS statistical model to prioritize possible contributors. So I just want to call your attention real quick to the left-hand column. So we modeled based on mass to charge ratio. And one of the reasons for this is that we're using ESI. So we're expecting that we're going to detect our peptides in multiple charge states. And that was really exciting in this case because we saw one peptide model in the top 10 likely contributors in 3 different charge states. So we thought that this was likely a strong candidate for contributing to activity. So this was the one we chose to follow up on first. And then just sort of as like a sanity check before we go down the full road of characterization, we check the elution of the charge states. So that's going to be the color traces in the front against the bioactivity profile in the back, just to make sure that the activity is actually tracking with the bioactivity. And it did. So you're ready to move forward with characterizing it. And in general, we take two paths. So we try to isolate for validation at the same time that we're trying to de novo sequence so that we can maybe have the peptide synthesized to validate activity. Because at this point, all we have is a mass to charge ratio. So today I'm going to focus on the mass spec side of that in our de novo sequencing efforts. And with Amaranthus tricolor, at the time when we started this project, we didn't have a genome or a transcriptome or anything like that to go off of. So we knew we were really going to have to do a full de novo sequence. And as I mentioned before, antimicrobial peptides can have a lot of different post-translational modifications. And as we start to consider more and more post-translational modifications, our potential sequence space just explodes. So we wanted to know as much as we could about the peptide before we started the sequencing process. So we used a variety of different sort of complementary techniques to learn a lot about our sequence. So we used chemical derivatizations, mass shift analysis, enzymatic digestions, and then some MS neutral loss analysis. And we were able to see that we had a disulfide bond, no acidic residues or lysines, multiple probably oxidative modifications, one of which was likely an oxidized methionine based on MS loss, and that we probably had a couple arginines in our sequence. So sort of as a result of pulling all of these methods together for this project, we ended up publishing just recently a review in Natural Product Reports to provide resources for other people that are trying to sequence antimicrobial peptides and get some more complementary information. So in the end, once we had all of this information, we tried a variety of different fragmentation methods to get sufficient fragmentation for de novo sequencing. And what ended up working was ETHCD. So with the help of Peak's de novo sequencing software, we were able to get this sequence, which I can show you right here. And it has a lot of the features that it has all the features we were expecting based on our orthogonal experiments. So we've got multiple cysteines that can form the disulfide bond. We've got our two oxidative modifications, no acidic residues or lysines, and we have a couple different arginines. So the last step before we worked with our synthetic collaborators at High Point University to have this peptide synthesized was to do some enzymatic digestions just to make sure that we have the sequence that we think we have. So we want to make sure that we produce the digest peptides that we expect. And also, we knew that those digest peptides would likely produce less complex fragmentation spectra. So we could resequence those chunks and have a really solid sequence to send to our collaborator. But really interestingly, when we did those digestions, we saw a couple different populations of the peptide emerge. So that hydroxyproline modification could actually be on 3 different proline. So it could be in the proline on position 4, 13, or 14. So the intact peptide that we were fragmenting, the non-digested peptide, was actually a chimeric spectra with multiple contributors. So we ended up having all 3 of those peptidoform sequence. And just also interestingly, the final sequence had really low homology to other plant antimicrobial peptides. So that's a strength of our pipeline, is that we're activity-based. We're not looking biased on homology. So we can find things that aren't what other antimicrobial peptides from plants sort of canonically look like. So we had all of these peptides synthesized, and the bummer is that they weren't active. So that was disappointing. But we had to kind of take a step back and look at our data and say, is there a reason that these aren't active, and what might that be? So we know that antimicrobial peptides can have some isobaric modifications that are really hard to detect via traditional mass spectrometry because they don't change the mass of the peptide. So they could have a D-amino acid, a cis-proline, or it could be the hydroxyproline, could be the hydroxyl group, could be at the 3 position rather than the 4 position, which is what we had synthesized. And we know that these can modify activity. So we wanted to look for some chromatographic evidence that there's a difference between our synthetic peptide and our native peptide. So what we saw was that when we fractionated both our crude extract and our synthetic peptide using strong cation exchange chromatography, there was a retention time shift between the native peptide and the synthetic peptide. So in purple, that's the native peptide, the elution of the native peptide across fractions. And then in blue, that's the synthetic peptide. And then if we assay the native fractions, you can see that the retention time of the synthetic peptide is in the less active fractions. So we have evidence that there's something going on with these peptides, and we're in the process of figuring out what that modification is. So that's kind of an ongoing work in progress. So with that, I, of course, want to thank my lab and all of the alumni. We've had a lot of undergrads that have put in a lot of work on this project, and we've had a bunch of collaborators as well. So Dennis Goldfarb and Ben Major at Washington University, the Womack lab at High Point, and the Shaw lab at USF, and our funding. So with that, I'm happy to take any questions.
Awesome. Great job, Tessa. Everybody please give her a hand. And so if you want to stop sharing, we'll let anybody that wants to ask her some questions. I know Jenny had a thought about maybe your inactivity with the oxidized methionine on there.
Yeah, so we have assayed it with and with, like, we've had it synthesized with and without the oxidized methionine, and we haven't seen any, it still hasn't been active, essentially. And we do see like lower oxidation states in our sample, and that methionine is always oxidized. So we're not seeing like it become progressively more oxidized with sample prep.
Have you tried, I assume you've tried putting all the peptides together as your bioassay, right?
Yeah.
Every combination thereof?
Yeah, yeah. And we tried kind of like spiking it into the native fraction that was active, thinking like, well, maybe there's just something else in there and then we'll see activity increase.
Yeah, these are high risk projects, high reward. They're,
yeah.
But well done. I'm sure you learned tons of skills. These are hard to chase down. It was super well done.
Thank you.
Yeah, so what is your ultimate dream job, Tessa?
Yeah, so I'd like to transition into industry. I'm looking at graduating at the very end of the spring, beginning of the summer. So that's kind of my timeline.
Okay, so you got a little bit longer, but it's always nice to get something hooked up so you have someplace to go too.
Yeah.
This is John Yates. Have you all considered the possibility that there might be some D amino acids in there?
Yeah, absolutely. So we've tried a aminopeptidase M digestion with the thought, because it'll cleave from the N terminus and then stop at any D amino acids. So I feel pretty confident that there's no D amino acids in the second half, but I can do a chymotryptic digestion to sort of make the second half of the peptide accessible to aminopeptidase M, so it can like keep chomping its way through, but the prolines stop it. So there's definitely the possibility that there's a D amino acid there. And we've looked at trying to answer that question chromatographically as well with the all-L synthetic analog, because you can definitely get a shift in retention time there. But
you can get the peptide synthesized with a D amino acid.
Yeah, just with the hydroxyprolines, there's so many different variants, and I can't chromatographically resolve the variants. So that's really limited us there.
Yeah, the combinatorics get to be big. The other possibility is to do amino acid analysis.
That's sort of been on our to-do lists for quite a while, but the mixture of peptides, the mixture of peptidiforms is really limiting us chromatographically. So I've had a really, really hard time isolating it. So that makes it more difficult for D amino acid analysis. But we've thought about like, well, what if you just saw any D amino acid, even in a non-pure sample?
So John Sweedler's lab up at University of Illinois.
Yep.
In Urbana does a lot of this with neuropeptides, and so you might reach out to them and ask them for some advice on
Yeah,
a quick and easy, maybe a quick and easy way to look for that.
Yeah, that's actually where we started looking at the D-amino acid protocol. One of their postdocs came and gave a talk at UNC Chapel Hill, and I was like, this is very relevant.
Tessa, are you only looking in the States, or you're considering other countries as well?
I would prefer to stay in the States.
Okay.
Mm-hmm.
Well, great job, Tessa. So everybody, please give her one more round of applause. And I hope you guys have time to hang around just a little bit over an hour to do a short breakout room. So we'll do a 10-minute breakout room. So you'll go about four minutes over the hour. And if you need to skip out, you can skip out of your breakout room. But I think it would be great to meet a few people. But Twitter's been a buzz the last about day and a half about mentoring. And so our question today is going to be, what's your favorite mentoring characteristic that you would like to implement if you're getting a new job or that you like implementing for your group? So please introduce yourself, tell people where you're from, what you're working on, and your favorite mentoring characteristic. And
so, Aaron, you should tell why Twitter's been a buzz.
Why Twitter's a buzz. Yeah, I guess I shouldn't just lead it like that. Yeah, there's a paper in Nature Communications that I'm not sure exactly when it came out. I'm guessing it came out just yesterday or so, that is really being highlighted as saying that female to female interactions are not as productive as female or as male mentorship. So, and it goes on both the sides of the female as the mentee and the female as the mentor. So it's definitely something you guys should check into, but it's somewhat very sad. But there's a lot of inaccuracies in the ways everything's being done as far as citations, and not the metrics might not be quite
Should not have been published.
yeah. So
I shouldn't say it shouldn't be published. It shouldn't be published in that form.
yeah, definitely. So but yeah, so definitely check it out so you guys know what's going on, because it's
maybe JPR needs to put together a special issue on female mentor papers.
Yeah, that would actually be pretty cool.
You can be the guest editor for that.
I was never mentored by a female, different generation, so unfortunately.
No, I wasn't either.
It would have been better, I think, if I had been.
But yeah, so I'll break you guys out, and definitely check out that paper. It's definitely a buzz on Twitter, and if you want to catch it, it's in Nature Communications. So I'm sure it's right up there. But have a great time in breakout rooms, and I'll see you guys back here at about 6 after the hour. There you go. 1 minute, okay? Actually, I can take you outside.
We left first.
Oh, I thought it was like, Aaron, you just blend in with the snow.
She's
Also,
back.
you know,
I had to go get my cheersing thing. I was like, I don't have any drinks to cheers to at the
Got a
end.
water bottle.
Oh, I thought you were about to put on an
lemon
elixir
here.
press there.
I was like, oh no, I have to cheers with a calculator.
Aaron, I was Olaf for Halloween this year.
Oh, that's awesome.
Yeah,
I love Olaf.
I have like an Olaf onesie. So
That is perfect. I want to get a Minions onesie, so I think I'm gonna do that next year for when I have to teach.
yeah, definitely.
Shouldn't
Awesome.
you be Steve Carell's character and make the students be the minions?
That would be better. I should be Gru. Oh, I hope you guys all had an awesome breakout room, though. And I think we had another amazing mixer. So thank you all so much for hanging out a little bit over. And let's do our cheers. I had to go get my water bottle to cheers everybody, but cheers to another great mixer. And we'll be back again in December with another mixer. And then we're planning a holiday party. So for the middle-ish end of December. So hopefully we can get you guys all online and do some fun stuff. But have a great rest of your day. And I'll see you guys in a couple weeks. And happy Thanksgiving. Almost.
Thank you so much. Bye.
Bye, guys, and great job. Great talks today.
Thank you. Bye.
Thank you.